One. Oh, it's pretty down the well, the laughing, isn't it? It's not.
No, it was
No,
a
they
bit
were
down
actually
the
there.
end.
Hi there.
Hi.
How are you?
Good.
Excellent. We're just going to have a bit of a chat here.
Yep.
How'd you get? When did you first decide you liked science, and how did you get into it? What were the early motivations, as it were?
Well, at school, I was always particularly good at science and maths. So naturally, I did sort of, want to go towards science for university. And then starting university in science, there were some key things that stood out to me that really interest me, particularly in cancer. When I found out that it's your own body's cells that make up cancer, and it's your own body that goes array and grows out of control, I found that fascinating. And so from that point, I really wanted to work on cancer.
When did you find that out?
Yeah.
How?
I was doing a medical science degree, and the lecturer was talking about different diseases and how cancer arises. And I was actually horrified to think that it's our own body and our own cells that cause cancer. And that just fascinated me as well.
So what did you do from there to further that
Yeah.
desire?
Yeah, so I did undergraduate medical science, and I always thought if I stay in science, I want to do something that will help with human health. And as I said, I had a really big passion in cancer. And so I decided that I would do postgraduate research. So I did my honours year on chemotherapy and my PhD in that same lab, looking at how chemotherapy actually works against cancer cells. Can we make them more selective? Can we make them work better? And so that's what I did for my PhD, and it was very rewarding. Beyond that, I realised that chemotherapy is not specific to an individual's cancer. So some people respond, others don't. You can also get side effects from chemotherapy. And that led me to go to Johns Hopkins University in the States, in the USA, where I really wanted to look at the biology of cancer. So how do cancers start? How do they get a blood supply to actually be able to grow in the person's body? And how do they spread to distant sites in the body, which is the real problem and the cause of mortality for patients?
You ready for this one?
Yeah, maybe.
That's right. How does chemotherapy work?
Different chemotherapeutics work in different ways, but a common way is damaging your DNA, so your genetic makeup, your genes, and they can cause breaks in the DNA. That means that you don't get any production of RNA and proteins. So essentially, when you damage your DNA, the cell will either die because there's a death response, or you won't be able to produce everything that you need to grow. So chemotherapy stops the cells from dividing and the cancer from growing.
But not every cell.
No, so some cells are particularly responsive to chemotherapy and others aren't. And there's a lot of research into why this is. We know some of it, we know why this happens, but there's a lot of unanswered questions. And interestingly, one of the reasons that chemotherapy may not work in some patients is because it actually relies on activating your immune response. So what a lot of people don't realise is not only does chemotherapy work by actually killing your cancer cells, it actually works by triggering a response so that your own immune system comes in and kills your cancer cells.
So breaking that into the two, I think immunology seems fair enough. You trigger a signal within your body, and the good guys come and kill the bad guys, basically.
Yeah, so with chemotherapy, you can actually trigger a response similar to if you had a viral infection or a bacterial infection. So the cell can recognize these, what we call nucleic acids, like pieces of DNA or pieces of RNA, your genetic code, and they recognize that they're where they shouldn't be. And in a oh,
Sorry again. Sorry.
yeah.
Clear my throat and block everything.
Yeah.
Your necklace is good. It's all
So chemotherapy can definitely trigger an immune response and actually make the immune system fight cancer, because it does this by breaking up pieces of your genetic code, of your DNA or RNA. So the cell recognizes this as different, similar to how it would see a virus or a bacterial infection. And this stimulates an immune response and causes the immune cells to come into the cancer and actually start fighting the tumour. Unfortunately, this doesn't work for every cell and in every patient. And we're trying to work out why some people respond this way and why others don't.
So you did your PhD in this and did research into it, and that took you so far. What was your next step?
Yeah, so the chemotherapy work was really interesting, but I really wanted to be able to treat people as individuals and look at the biology of everybody's cancer, how it's different, how we can use that knowledge to really personalise therapy for every patient. So that took me to Johns Hopkins University, where I worked in a breast cancer lab, and I was specifically looking at how cancer changes as it grows in the breast and then how that looks different to when it grows in another site, such as when it spreads to the bone. And what we found is it's a constantly evolving disease. So even in the same person, the cancer can change and look different depending on where it's growing.
So is it then the same cancer? It's changing. It's hiding.
Yeah, so not only do many people have different types of breast cancer, because breast cancer is not just one disease, it's quite different in different people. But even in the same person, breast cancer can look very different from where it started in the breast to when it spreads to other sites. And even within those sites where it's growing, it can look very different depending if it's growing near a blood vessel, whether it's near an immune cell. It changes its properties. And so we really need to bear this in mind when we're trying to target cancer, that it does change.
It's a one type of cancer that is more proliferates more than others, or we just established there's so many different types of cancers. It depends on your body type, doesn't it?
Yeah, well, cancer can proliferate at very different rates. In fact, a cancer in one individual can be proliferating faster in one area of the breast than another area of the breast. And it can depend on many things, including if there's a blood supply nearby, the biological changes in that cell that allow them to keep dividing, whether they've spread to a distant site and they're trying to change what we call the microenvironment in that site to promote the cancer cell's own growth. So even within a patient, an individual patient, the proliferation or growth of their cancer can be different depending on the site of growth.
Wow, what do you do about that? Well, first you've got to find it, right, and figure out why it's doing that.
It's very difficult actually, because chemotherapy does work quite well against cancers that are actively growing and proliferating, and sometimes when cells have actually what we call disseminated or moved to a distant tissue such as the bone, they can be sitting in what we call a dormant or non-proliferative state, and that means they're sitting there and not actively growing, and these cells aren't particularly sensitive to chemotherapy. So we really need to find a way that when we do treat patients that are diagnosed with breast or other cancers, that we're not only removing the primary cancer, we're also targeting those cells that have already escaped.
So they've gone deep cover, basically, and they're just to be scary, they're waiting their time.
Yeah, so over 50% of breast and prostate cancer patients that are diagnosed might already have cells that have spread throughout their system and could be lodged in the bone, for example, in the bone marrow. So they're just sitting there in almost hiding in a sort of a dormant state where they're not actually actively dividing. Now, in some patients, they can sit there and not become problematic. But in others, they start growing, and that's where cancer becomes a real problem, targeting those cancers that are starting to invade these distant tissues that are associated with the mortality in patients.
And if it's already there throughout the body dormant, if it takes off, it goes lickety split.
Yeah, so, we can look at the primary cancers, and that tells us a lot of information. But ultimately, to cure metastases, there are two things we could do. One is we prevent cancers early or detect them early. And that way we're targeting cancer very early on before these cancer cells move throughout the body. The other is we get better at targeting them once they've spread, once they become a completely different beast to the primary cancer.
What do you love about your job?
I really find my job fascinating. The fact that we can ask questions that no one has asked, but actually ask these questions that may be difficult but could make a real impact for patients. Like being in a place like Peter MacCallum Cancer Centre, we see the patients coming in for treatment. We can see when they're not doing well, when they're unwell. And what we want to do is change that. We want to be able to give people peace of mind. This is the type of cancer you have. This is the therapy that will work for you. You have a fantastic survival rate with that therapy. So psychologically, they can go home with that knowledge about what they have and that they're getting the best treatment for themselves as individuals. And that really drives me. It makes me passionate about my research, is that I could influence many people that do develop cancer and their families, because it's extremely stressful.
How far has it come in 20 years, though? It's more.
I think
Keep you laughing.
since I've been doing research, one of the biggest, changes has been the use of immunotherapy. So using your own immune system to fight cancers, and certainly in things like metastatic melanoma, we've seen this go from a disease that could take most of those patients' lives to actually seeing almost a cure in up to 50% of patients. This is remarkable. I think we've only just started with immunotherapy. I think we can get immune-based therapies to work in more patients. The more we understand the biology and the immunology at an individual patient level, I think we can actually get immunotherapy working for more people to protect against the really deadly types of cancers, the metastatic cancers.
Tell us about the really deadly types.
Yeah. Okay, so could I talk about bone metastasis because I have a particular interest in that, or any?
Simplify the term, or
Okay. Oh, yeah. Okay. All right.
Because I sort of understand, but
Yeah, sure.
I'm a cookie.
No. So if I use the term and I explain it, will you then use it first with it explained, and then can I use it later?
A subtitle.
Oh, okay. All right. Yeah. I just have to know if I have to say it every time, explain it every
No,
time.
once you do it once, that's
Okay.
we're good.
Yeah. The problem with cancers is, when a primary cancer arises, whether that be in the breast.
Mad big fun. There's an alarm. Thanks for the alarm. Be afraid.
I didn't even notice. That's problematic.
Now, what we're trained to hear alarms.
So the problem with cancer is, upon diagnosis, we can remove cancers from a lot of their primary sites, such as the breast, the prostate. But the issue is when it spreads to distant tissues, such as the lung, the bone. In fact, bone is one of the most prevalent sites of cancer spread for solid tumour malignancies. This is problematic. The cancer, once it metastasizes, the survival rates decrease to about 20%. So we just are not doing good enough at curing metastatic disease. So did I say when it spreads, or did I not explain it?
You do it again.
I might have to do it again.
I think so.
Yeah, okay.
Look at that mum.
Yeah,
Does it actually sit on the bone or in the bone?
inside.
So it's simply that.
It's actually inside the bone. It eats away at the bone, and it actually harnesses those the bone cells to help it. So the bone cells eat away at the bone, and it releases growth factors that help the cancer cells grow. So it's called a vicious cycle in the bone. Yeah.
So once started, it just keeps on.
Yep. So the cancer gets there. It stimulates what we call angiogenesis, which is a blood supply, so it can start growing. Once it starts doing that, it recruits the bone cells, starts degrading bone or causing abnormal bone growth. That releases growth factors. The cancer grows bigger and does that more. So people with bone metastases or bone spread will often have terrible pain because, they could get bone fractures. It's quite a morbid disease, and it's very difficult to stop that vicious cycle.
That's bloody too good. I can't help that.
No, no, but just ask that last question. Where is the cancer inside the bone? Is the tumour in the bone?
Why does it metastasize? Like we talked about where it goes, what triggers.
I was going to get there.
Okay.
I was going to face it. Those bloody turncoats of the Pope. But I think Steve wants us to start again on that.
Okay.
Just another question about where the tumour is.
And then we can follow it through as to why.
Okay. Well, if we knew that, we no, but we know some of it. Sorry, can you ask the question again?
I've forgotten the question.
Oh, good. So have I.
Thanks for saying that.
It was
Look, where's the tumour?
Yes.
In the bone cancer, where is the
How's
tumour?
yes.
You said it's inside the bone.
Yeah.
Okay.
The tumour. Well, I said, does the tumour form on the bone? And
There is, that's it.
That's what took us into that labyrinth of fun that we had together.
yeah.
Does a tumour form on the bone?
The tumour that spreads to the bone often forms inside the bone, often within the axial skeleton. And this is very morbid disease because the cancer cells grow, they affect the bone cells themselves that can degrade too much bone or form bone where it shouldn't be. And this is extremely painful for the patient, but it also means that the cancer can continue to grow more. It utilises the bone environment to actually support its growth.
you did say it sort of recruits the bone cells to help. And I was going down that bloody turncoats. How does it do that? Again, we might not know, but it does it.
Yeah, I mean, the cancer cells release particular what we call chemokines and cytokines, so signals that come out of the cancer cells that can recruit other cells in the bone to help them. So this could be a type of immature sort of immune cell that can help them, the bone cells themselves. So when what's called an osteoclast comes into the site, it can actually degrade bone, release growth factors that help the cancer grow further. And that can cause huge pain for a patient because if you have too much bone degradation, you can get fractures. The same with bone growth. When you have abnormal bone growth, this can cause huge pain for a patient. So the ability of cancer cells to recruit other cells and then utilize the bone cells to support their growth is a major problem that's associated with the pain in bone metastases.
I can't even imagine. But there's no nerve endings there, so they must be so deep-seated.
There are nerves that are
And on the bone.
established. Well, there are nerves.
Attached to.
Cancers are. It's quite interesting. There are nerves, and neural filaments and things like that that are associated with cancers, and that's a whole area of study. And some of them can support the growth of cancers. So there's a whole programs of work around stress, nerves, neural networks, and cancer.
Tell me a bit more. Now I'm intrigued.
Oh no, it's not my area.
Tell me.
Well, I guess, yeah.
So did you say there are nerves inside tumours?
Well, there's neural networks. They actually have found the markers that are, of nerve cells and associated markers inside cancers.
Was this unexpected?
I think originally researchers were looking at this because they saw an association with a stress response and an increased risk of spread of cancer. And so that made them look into these networks closely. And it's not unless you look at this and look at the biology that you actually find out, how it works. How does metastasis occur or the spread of cancer occur? What other cells are involved? Cancers do not progress by themselves. They utilize so many other types of cells in the body. The interactions between a cancer cell and another cell is critical in whether it spreads to a distant site, whether it responds to therapies, including chemotherapy, and what type of cancer and what sites it will spread to. So actually, the cancer cell does not work in isolation.
And stress is affected.
Yeah, so studies, for example, in breast cancer using different models have shown that the actual ability of a cancer to spread after a diagnosis of primary disease can be enhanced by a stress response. And if you block that, you can actually reduce the spread of cancer.
Wow.
Yeah, but that's not my work.
But you know about it, right?
Yeah, it's quite interesting.
Yeah.
At the same time, I don't really want to alarm patients that it's just another thing they have to worry about.
Yeah, sure.
They can't drink alcohol. They can't be stressed. If you tell someone they can't be stressed, it's going to make them
What's
stressed.
the first reaction there?
Yeah. So it's one of those things that sometimes these things are helpful, but other times it's actually difficult for a patient to have all this knowledge about what they could have done wrong. Usually it's bad luck. If a cancer arises and spreads, most of it is that the person was unlucky. It wasn't something that they did.
Yeah, and saying learn to relax always works.
Yeah.
Just like saying calm down.
Yeah.
That always works too.
It's like telling me not to sweat from the light right now. Yeah.
Ben, would you like to ask a few questions? Do you want to come and sit down?
I think you were going to ask about how
I had lots of them.
I was going to leave.
you were going to say how does it actually spread to the bone? Yeah.
I mean, if the answer is if only we knew, fine. But
Well, you can ask the
because
question.
I know the blood supply, like certain things have to happen for it to grow, and then presumably certain things have to happen for it to spread.
Yeah.
I'm also like the dormancy is really interesting too, because if you could suddenly make everything dormant, you wouldn't have to
Yeah.
worry
So
about
there is an idea. Can you push something?
Actually, if you look at me,
Yep.
you can answer the question.
Yeah.
Look at me.
Yeah. Dormancy is a really interesting area. So, cancers can spread very early before their detection. and they can be sitting in a distant tissue in what we call a dormant state, so they're not actually actively growing. This is very difficult to model in the laboratory. You know, everyone may be different. Why is that cell sitting there like that? How can we keep it in that state and prevent it from growing into a large cancer in those tissues, such as the bone? And there is a lot of work in this area. In fact, these cells are also quite resistant to chemotherapy because they're not growing. So there has been two ideas. One is, can we target these dormant cells? And it is thought you could target them with an immune response because these dormant cells still have the signals on them that would allow them to be recognized by the immune system. So that's one idea. The other idea, can you bring them out of hiding so that they are targeted by chemotherapy and things like that? That's a risk because you're bringing them out of a silent state and you're allowing them to grow. And so ultimately the best thing would be to either diagnose cancer early enough that they haven't, leaked out into the circulation and lodged in distant tissues, or that we could go and mop those up and target them. And I think immunology, may hold one of the important keys in doing that.
Give the chair a power.
I think one of the problems is you may answer the question, but we may not understand time. So just going back to that, so a friend of mine had prostate
Yeah.
cancer, and it had spread beyond the cap or something.
Mhm.
So he said, so I don't really know what that meant. But what this no, because we hear about people who've got stage 1 and got cancer or something, and then it always, the fear sets in when it's spread.
Yeah.
So, and it was fascinating too before you were saying that they're completely different cells.
Yeah.
So, what's happening? What's going
Yeah.
on?
My laboratory has been really interested in the process of metastasis, so the spread from a primary cancer to a distant site. And the reason is because we can treat primary cancers very well, whether it be by surgery or other therapies. But once they spread or metastasize to distant tissues, such as the most common site, the bone, it's very difficult to target them. And one of those reasons is because they become very different to the primary cancer. What we've found is that the immune system is quite important in this, that the cancer cells become invisible to their recognition. And so once they've spread, it's very difficult to target them, whether that be by an immune-based therapy, where we want the immune system to attack them, or by chemotherapy, because actually a lot of the therapies we use today to treat metastases are therapies that need the immune system to work.
You make me think of alien in that. So like, is the first cancer is like the alien in pods. It's growing there, but then it comes out of the pod and it gets really scary and it has acid for blood and stuff. So
Yeah.
you're dealing with a different foe. Is that what?
Yeah, I mean, some of the properties of the primary cancer, there will be some cells in there that have a better ability to invade out of that site, whether it be the breast, prostate, or other site, and enter the bloodstream. Those qualities could have become apparent because of the interactions between those cancer cells and surrounding cells in what we call the microenvironment, so the environment that the cancer is living in. Now, some of those qualities will be very useful for spreading into the bloodstream, but the cancers may need to develop other qualities that are completely independent to survive in another tissue, such as the bone. And we know that. And the reason we know that is because metastasis is not actually an efficient process. It's efficient to get cancer cells in the bone marrow, for example, but the step between having a cancer cell lodged in the bone marrow to growing into a large cancer in the bone and causing issues clinically, that's quite inefficient. So we know that over 50% of patients could have cells growing in their bone, or sitting in their bone, sorry. I'll say that again. So we know that upon diagnosis of breast and prostate cancer, at least 50% of patients could have cancer cells that have already spread beyond the primary cancer through their bloodstream and into the bone marrow. But what we do know is that many of those patients will not have deadly metastases develop. What that means is that there's this interaction between cells and their surrounding microenvironment, so the cells around them in the tissue that they reside, that allow them to develop the qualities that they need to develop into large cancers that are problematic. And so those interactions are critical in the ability for us to find new therapies to block that stage, which is the deadly stage.
So you mentioned cytokines before. I was just looking at abstracts from, I think, about cytokines. Sounds like there are good cytokines that help fight or muster the response, and then the cancer produces cytokines too to stuff you up. Is that
Mhm.
talk about that?
Yeah, so cancer cells secrete proteins, whether they be cytokines or chemokines. Now, these can be good for us. So, for example, we've found that the production of one type of cytokine called type 1 interferons that are really important in protecting us against viruses and bacterial infections, for example, they're also really important in protecting us against cancers. What happens is the cancers that are growing in sites like the bone can completely hide from being, recognized by the immune system by blocking these cytokines. So they actually turn off the ability of the cancer cell to actually secrete or pump out these proteins, and it means that they're invisible. So they're the good cytokines that we want. What they then do is secrete or other cytokines and chemokines that bring in the bad guys. And that means that they bring in cells that help them survive, that block the immune response, that help the blood vessels be recruited. So there's a real balance between the pro or the good cytokines and the bad ones.
So, I mean, it's just growth without control. Just everything seems to be just going mental. I'm sure it's not a scientific term.
Yeah, it's interesting because a primary cancer has already developed the qualities it needs to grow. It's already overcome the balance that we usually have with our cell death pathways. But when these cells spread to a distant tissue, they often need to develop another quality as well. And what we find is that quality is actually avoiding recognition by the immune system. So they've already got the quality of being able to grow, and replicate and become these large cancers, but they also need to block particular interactions between the immune system so that they can actually survive. And so it's quite interesting that it's not just about whether a cancer can grow, it's also about whether it can survive other challenges on its journey from a primary cancer to a metastatic cancer.
And you're also famous for like developing tools. Is that fair to say?
Yeah.
I mean, some of the things articles in you seem to be about breakthroughs in because cancer does seem to be about brains and tools and lots of other things. But
Yeah.
tell us about some of those developments.
Yep. I would say my most exciting findings, so the findings from my team, have come from multidisciplinary collaborations with other experts. For example, we have a real interest in two areas. One is we're diagnosing cancers a lot earlier. We have to make sure the diagnosis is accurate and that we're not over-treating patients. And then at the other end of the spectrum, we have these cancers that still escape and become metastatic and very untreatable. And so we're trying to target those. So for the two ends of the spectrum, we've developed new shivers. I hope you can cut the first bit and then put this bit back. Okay.
We can.
Where will I start from? The multidisciplinary that I can't say properly. Do people know what multidisciplinary is? Like the public?
I think so.
Yeah.
I wouldn't bank on it.
Okay.
I would play it safe.
Okay.
Can you explain it?
Yep.
Like it's a collision of
Basically, different areas coming together to solve a problem.
It says what it does.
Yeah.
But it's like,
Yeah. What fascinates me about cancer research is bringing together different disciplines to solve a really critical problem. And recently we've done exactly that. We collaborated with physicists and developed a new slide technology that is actually a stain-free technology that you can put a tissue on that's been removed, such a biopsy from a patient, and we can actually pick up cancer cells differently from normal cells. And this is incredible because it's really useful to help a pathologist diagnose whether someone has a cancer at a very early stage.
why, like the old-fashioned way would have been using a stain. So is this why is this better?
Yeah, so usually when cancer is diagnosed, a pathologist looks under a microscope and looks at a piece of the cancer on a slide that has been stained with a purple and pink colour. So they can look at the shape of the cancer cells, they can look at how the tissue is actually organised. What we've developed in collaboration is a slide that doesn't just show you what the cells look like, but also tells you by a colour distinction whether it's a cancer cell or a normal cell. So the cells actually light up a purple sort of green colour, whereas the normal cells stay yellow. So if a pathologist looks at this slide under the microscope, their eye will be drawn to the colour change. And we really feel like this will aid in early diagnosis when we're dealing with small cancers that are picked up very early and can be missed.
So if you didn't have that colour change, you'd be having to look at the cell shape, other things that are harder to spot.
Here at the moment, pathologists look at the cell shape, the organisation of the cells, if something's out of place, and they do rely on that. And we've relied on this for years. Diagnostics is still pathology driven using a stain called hematoxylin and eosin staining. So the slide that we've developed, you don't stain it at all. You take a same piece of the cancer, you put it under the same microscope, but there's a special coating on the slide that allows you to see different colours depending on the properties of the tissue that you're looking at. And we can actually see these cancer cells light up differently. So what we think is, in the future, this slide would be a really good adjunct to the normal assessment, so that a pathologist not only looks at the morphology, but can be really confident, is that a cancer cell or not? And we feel like this would be really useful in a number of cancers.
Right. So, yeah. So it's fascinating how one thing spoke to Ricky before, we were talking about what goes on in Peter MacCallum is you've got all these different areas going on. Talked about a recent paper that was done with Doherty's.
Yeah.
And how it was the sort of meeting of minds. It was like people having a chat. So how on earth did you get hanging out with the physicists?
Yeah, so knowing that multidisciplinary research is really critical in making the best discoveries, there was an initiative where we took different streams of research and came together in one room. And from doing that, we started discussing, how we could bring our fields together. And so 8 years ago, I started working with Brian Abbey from La Trobe University, and we came up with this idea of using his sensing expertise with my cancer biology expertise to actually see if we could develop something that could be utilised in a pathology space. And it was incredible.
I mean, it is fascinating how, because research by its nature means you need to be so utterly focused on what you're doing that you can kind of not know what everyone else is doing. So how did you know each other?
Yeah.
How did that come about?
What I really love about research is the people that you meet, the ability to be able to say, I don't know everything. I'm not an expert at everything. So I know what my laboratory's expertise is, and sometimes we'll have a difficult question that we just can't tackle ourselves. And so really, it's really fantastic to be in the Parkville precinct. We have experts that we can reach out to and collaborate. And also, this doesn't sound good. I've actually got a better example for the bone scaffold. I don't know.
We don't know anything about that. So
Yeah.
tell us.
And then you can tell me if you want me to do the question differently. Yeah. So a really exciting project that has been so far, really successful is the collaboration between my laboratory in cancer biology, engineers at Melbourne University, and clinicians at the Peter MacCallum Cancer Centre, where we've been able to develop a system where we can take cancers growing in the bone of a patient directly from the patient at the time of death and grow them on a special bone scaffold to allow them to stay alive, so we can test therapies that work in our models on the actual individual patient's cancer. And this has been quite remarkable because the engineers have designed what we call a microfluidic chamber to allow them to grow in the right environment. The clinicians have been able to recruit patients that are very happy for us to utilise their tissue to test this, because this could be a remarkable breakthrough for personalising therapy for patients. And so bringing those teams together allowed us for the first time to grow cancers from bone out of the patient to test therapies, and we're really excited about that. Did I say that funny?
I understood it. Did you have a positive
Oh.
Yeah.
It's the second time around.
I.
But the scaffold, it's sort of.
It's actually a cow bone too, so it's quite
that was
interesting.
the question,
So I went into the butcher. I went into the butcher near my house, and
So you forgot the butcher.
yeah, I should have brought the butcher into it.
Stand down, code yellow. OzLab outage has now ended. We have resumed normal operation. Attention staff, stand down, code yellow. OzLab outage has now ended. We have resumed normal operation.
Because we love stories. Like, what, when we're talking about, I mean, everyone knows Archimedes and Eureka and whether it's true or not, but then Watson Crick used to go to the Eagle Hotel in Cambridge and when they were doing structured
Yeah.
DNA, and that played a major role. So a butcher is a really good
Yeah.
angle.
Yeah, yeah. Well, this probably the most exciting project that we are embarking on at the moment is really a collaboration between engineers, my laboratory, cancer biologists, clinicians for patient samples, but also the local butcher. So what we've been actually able to do is take samples from patients at the time of death, samples that don't usually grow outside of the patient, and these are cancers that have grown in the bone. They're very difficult to keep alive, which means that it's really hard to test if therapies that you've identified will actually work at an individual patient level. So this collaboration has allowed us to take these tissues, put them on a cow bone scaffold, the cow bones which we got from the butcher, a scaffold and a microfluidic system to grow them in that was the engineer had designed. And we've been able to keep these cancers alive so we can test new therapies. And we're hoping to develop this into a pipeline of precision medicine for patients that develop bone metastasis.
So precision medicine seems to be something we hear about a lot. Is that,
Just hold that for a second.
yeah,
So you're keeping the cancers alive so you can kill them.
So we're keeping them outside the patient. So ultimately what we want to do is so when a patient is diagnosed with bone metastasis, is to take a biopsy of that cancer, put it on our system, our bone scaffold system, and actually test if a therapy would actually work for that individual patient. So we want to actually tell them what therapy will work, tell the clinicians what should this patient be receiving, how can we give them the best chance of survival, and how can we make it specific to them as individuals.
so the bone scaffold at the moment is being used with the cells from the dead people, but in the future it would be a live biopsy that you could grow in testing. Is that right?
Yeah, so at the moment, we're utilising tissue taken from patients at the time of autopsy with the ultimate goal that we could take them from patients that have just been diagnosed with bone metastases and guide their therapy. And we know we can do this. We work currently with clinicians at Peter Mac, including Michael Hoffman, where we can take bone biopsies from patients and actually look at their cancers individually to try and determine what they will respond to. So we really feel like this could be part of a new pipeline of personalised medicine.
How did you get the money? Because it sounds like
Oh.
going in there and saying, okay, I've got this plan. I was talking to the butcher the other day, and how did you go about raising funds for that?
When we had this idea of, how are we going to grow these cancers from patients with bone metastases, we thought, how are we going to get funding for this? It's a big risk, but if it works, it's a huge gain. We were lucky at the time because the Peter Mac Foundation supported us through a Peter Mac Foundation grant. And it's often philanthropy like that that can lead to the big, big, risky projects that actually can lead to a cure, lead to the, huge benefit for a patient. Many of the standard granting, funding agencies, it's quite difficult to ask a, hugely new question that you don't have any preliminary data for. That's where we need, these donations are critical. They're actually giving us the ability to ask a brand new question in a totally new way, and it could lead to something really big.
And is that your experience with the Australian Cancer Research Foundation? Can you tell us
Yes.
a little bit about that?
Yeah, so it actually relates to the bone project. Yeah, so when we were designing the project to actually grow bone metastases on this cow bone scaffold, we realised we need to be able to look deep inside the bone to actually see what do the cancer cells look like? How are they responding to therapy? Can we actually use fluorescent markers to see how are they changing. And we knew we needed state-of-the-art equipment. State-of-the-art equipment that we didn't have, and that it's really difficult to purchase because there is no funding mechanism for it. We were really lucky that the ACRF supported this. It's very unique. The ability to buy, the best state-of-the-art equipment is critical to fund research that is asking really important questions. And that was where the ACRF was amazing. We were able to purchase this equipment, and now we have an opportunity to really test whether this new technology and this new scaffold system is going to make a difference for patients.
Do we have to say Australian Cancer?
What do I have to say? Australian Cancer Research Foundation. Okay.
Are we shooting the equipment?
Well, I think the rest is it. Well,
Can we?
do you have the equipment?
It's not here yet, but the facility, they wouldn't know. Like the facility has some really nice. So one of them is the microscope that we're getting, but we're getting a much better one. The other one's going to be decommissioned, but it looks good. Like, you wouldn't know. But it's not the one, so just make sure you can't see the logo of the company.
We were really
Okay.
fortunate that the Australian Cancer Research Foundation and do the NAS, but you come to that and discuss the money together.
Okay, I'll try and jump up and down. But it actually is true. We wouldn't have been able to get it otherwise. It's completely true. We were extremely fortunate because we were able to get funding from the Australian Cancer Research Foundation to support equipment that is critical for our new bone scaffold model. So, we've developed this fantastic new model for looking at bone metastases, but we need to look really close at these cancers, see how they change with different treatments, see if we can use fluorescent markers to look at this dynamics and interactions with other cells, including immune cells. And so the state-of-the-art equipment that we really need, we had no funding for that. And the funding mechanisms for that are really tricky. It's really hard to get funding for state-of-the-art equipment to ask, some of the most important questions in research. And so that's why it was critical for us. And we were, really fortunate to get the funding from the Australian Cancer Research Foundation.
If you had $100 million, what would you do with it?
If I had $100 million, there's a few things. Supporting the best state-of-the-art infrastructure, because you can be as smart as you want, but you really need to be armed with the best technology and also the best resources. So I would also use it to set up better biobanking. So we have a huge resource at Peter Mac with patient samples. Our patients do want to give back to research and help, other patients that are going through what they have. And so the ability to actually set up new models using human tissue and actually utilise this alongside exciting projects, and personnel to test the next big questions, including growing these bone metastases and actually trialling new therapies for personalised medicine, I would definitely use the funding for that.
So,
I think I said that really badly.
but no, not at all.
Oh.
But I'm just, I think, to me, the most interesting thing is this collaboration.
Mm.
Like, and just setting up an institute where, because it is physicists.
Actually, I might say that.
This is all Peter Mac.
A multidisciplinary hub.
Yeah, why not?
Yeah.
Or,
It sounds pretty grown up.
or yeah, or like what's the next big, what's the big bet? Like two questions really. What would you do with that money? I mean, they're related, but what? When we're not going to say where's the cure going to come from. That's naive. But what are you most excited about? How are we phrasing this?
I think.
What are you most excited about? Well, this is the $100 million question.
Do you want me to talk to the $100 million question again?
I made it up. I don't know.
No, it's all right.
I could spend 100 million pretty quickly, to be honest. If I was given the opportunity of having $100 million, I need to start that again. If I had $100 million, I would definitely support these multidisciplinary projects, bringing in engineers, physicists, clinicians and scientists and the best infrastructure to actually work on a problem that you can solve as a group. And the bone metastasis work is a perfect example of that. Funding people to come together and to organise a project that they would never have been able to do in isolation.
The interesting thing there is, like, if we don't really know where the big breakthrough is going to come from, it probably is more likely if you've got a whole bunch of different people doing it.
Yeah, I mean, we don't always know where the next big breakthrough is going to come from. I mean, we all have our own ideas and, we're trying to follow those ideas. But when you bring people together and you have the conversations and you start doing this really exciting research that requires a multidisciplinary approach, that's where you find the next big thing. And that's where you do make changes.
We talked a lot with Ricky about teams, and Sandra spoke to us
Mhm.
about it last year, this whole thing about cancer research as a team sport.
Oh, yeah.
Which was, but the interesting thing about having engineers and they wouldn't have a clue what you're talking about.
No.
And so
And I don't have a clue what they're talking about.
Exactly.
It's the Big Bang Theory all over.
It's fascinating being in. I just spat. I saw it. Shoo. I'll have to do that again. It's fascinating being in a room with engineers, physicists. Like, we have a discussion and we bounce off each other because we really have little idea about what each person is actually discussing. But when you come together and you discuss it, it becomes clear what you can do together, and it's quite fascinating. A common theme that you can find, that you can ask fundamental questions about that you wouldn't have previously. That didn't sound good. Oh, really?
Yeah, well, you were absolutely engaged,
Like-
and you're excited about it.
I am excited about multidisciplinary research.
What's exciting about it?
The one thing about my career is I've never, I guess, what was I going to say? What I've really loved about my career is I've never put my hand up and said, I know everything. Okay.
Okay.
One thing I love about research is that, you don't need to know everything. You can have an area of interest, you can have expertise, but it's the ability to reach out to others to ask something you wouldn't have before. And I love that, being able to work with people with completely different minds and come together to solve a problem is so fascinating.
Well, if you ever need help from an advertiser, love to help.
Oh, yeah. That'd be awesome. Like, we're actually talking because I also work on looking for markers in the cells surrounding the very earliest stages of breast cancer to actually tell you if you get diagnosed with early breast cancer, could you just be left alone or have very minor surgery with no radiotherapy? Because at the moment, women are diagnosed with these early stages of breast cancer. They think they've got full-blown cancer. They're worried they're going to wake up and get an ache in their bones. They think that the cancer's come back. It's constant stress for the rest of their lives, to be honest. They feel like they're just waiting. That's what some patients have told me. So what we're trying to do is actually develop markers so we can say, you don't have that type of cancer. Let's give them the peace of mind that it's okay. If your surgery is all you need, you can live your life to the fullest. So what we want to do is Bruce Mann is from the Royal Melbourne Hospital and he's a breast surgeon. We want to actually make something that's visible to the public to know that actually most of you will be fine. Because when they're diagnosed with early breast cancer, they think it's a really bad thing. They think they have full-blown cancer, but they don't. In fact, 95% will be fine. So if we can add confidence to that with our markers, we can literally say, you're fine.
So you need the equivalent of polyps.
Yeah.
Because cancer, the problem is the label.
Well, polyps, exactly. So polyps is very similar. I would say ductal carcinoma in situ, which is the early stages, they are cancer cells inside the duct. But with surgery alone, 90-something% will never go on to get distant spread. Oh, sorry, I should have said that properly. I forgot we're just not having a conversation. If I said 90-something%, that's going to sound really good. I'll say over 90%. Yeah. So what a lot of women may not realise is that when they're diagnosed with early breast cancer, called ductal carcinoma in situ, most of them, over 90%, will never have their cancer come back in distant tissues like the bone, the lung, the places that will actually be problematic and take their lives. So they're living thinking they've got breast cancer. They're worrying that the cancer is going to come back, but most of them will be fine. What we need to do as scientists is give them that peace of mind.
And that would be the stress kicking in.
And then they get stressed because they're worried that their cancer might come back. And stress is not what you want when you are going through a cancer diagnosis. Being stressed probably isn't the best thing when you're talking about response to therapy and your risk of the spread of your cancer. So, the more information we can give patients or people with cancer about their specific cancer. Are they receiving the best care possible for them specifically? Are they likely to have a problematic cancer that comes back later on in another tissue? And can we treat it now to prevent that? We need to be able to arm patients and their families with knowledge, and that's where science is critical.
What's the current death rate from breast cancer in Australia? Is it high?
No, well, it's still breast cancer is still the second leading cause of cancer-related deaths in women because it is such a common disease. But over 85% will survive upon a diagnosis of breast cancer, and those statistics can change a little bit. But so primary breast cancer is very treatable, but it's so common that the remaining patients that do get spread of their cancer, that's a lot of people. It's still the second leading cause of cancer-related death. And often I hear we should be funding the really deadly cancers more. That's true, but breast cancer is still a really deadly cancer because it's so common. So it's still the second leading cause of cancer-related death. So, we still have to do more for breast and prostate cancer, given their frequency, and that we still cannot target metastatic disease.
That was really good.
Yeah.
You want to tell her?
Huh?
That was really good.
Oh, was it?
That was really good.
Oh.
But how long do you have to dash?
No, what's time? No, someone put it in my diary till 4:30, but I don't need to be here till 4:30.
No.
You can tell me to go. Yeah.
We like you, Sue.
But it's one thing I'm intrigued by, and Kayleen talked about this, like you talked about collaboration with engineers and butchers and things, but what about other cancer research institutes in
Oh, yeah.
Australia and overseas?
Yep.
How does that work?
Yeah, so we collaborate a lot within Australia and internationally. So, for example, in our projects looking at finding ways to predict whether a patient with early breast cancer will go on to develop a problematic type of cancer, we utilise patient cohorts from all around the world. We utilise pathologists so that they can actually look at our findings and we make sure they're reproducible findings. It's to think about something else. There's a few things, I guess. Collaborating with people nationally and internationally not only gives you different expertise to come together and ask the question, but it also gives you access to multiple cohorts of patients. So when you have a finding, you want to make sure your finding is relevant, not just to someone in Melbourne, but it is nationally and internationally relevant, because you really want to drive this finding to make an impact internationally.
Has the personalised medicine, you spoke to
I'm omnipresent.
ProCan, was it ProCan that you spoke to? Someone from ProCan? Their whole thing is a
Building up
tissue
the bank.
data bank.
Yeah.
Exactly.
Yeah, so that seems to be
Same with the
happening.
clinical trials.
Yeah.
Like, if one clinical trial gives you a response rate of, 20%, was that a one-off? Was that the nature of the patients in the control group versus the treatment group? You really need to do these trials worldwide so that you can really make an impact and actually determine, is this treatment going to be something that becomes standard of care for patients with a particular type of cancer? The other thing with precision medicine, what really gets to me, is that we're getting a lot better at treating, for example, women with breast cancer. We're treating them before surgery. And we're getting a lot better at increasing the survival rates and targeting therapy. But we're still not good enough at saying, okay, that person will respond and that one won't. We can say, for example, 60% of patients will, but am I in the 60% or am I in the 40%? And so what we still need to do is actually be able to tell each patient, are you likely to respond to this therapy, or should we put you on something else that is likely to work for you as an individual?
And how far off is that?
Oh,
Genuine precision
yeah.
medicine?
I
Really
think,
close?
yeah, I think in the future that's what we're going to get better and better at, personalised medicine. You know, looking at each person's cancer and utilising the best therapy for them. And that's coming from molecular screening, it's coming from the ability to actually track cancers in real time, whether that be by imaging or other, you know, technologies. Okay. Okay. So I think what's really exciting for the future is, you know, precision medicine, the ability to treat each patient as an individual. We've got better with our technology for actually looking at the differences in cancers, so interrogating the cancers at a deep, deep level. We've got better at developing therapies that are actually working for more patients. What we need to do is link them together. So we can actually determine, are you the person that will likely be in, you know, the category of responders to a particular therapy? I don't know.
I can't remember anymore.
Have you ever had any eureka moments?
You take the seat.
Well, I got the
Have you
Eureka
been the
Award.
first one to
We
see
got the
something?
Eureka Award on the slide technology. I must say, when I first looked at the slide technology that the physicists and now group developed together, I was astounded. The fact that I could put a tissue on a slide, I hadn't done anything scientifically to it, really. I just put a section of tissue like you would when you know, do a biopsy on this slide and look under the microscope, and the cancer cells just came up at me. And I couldn't believe it. It was a really fantastic moment that, you know, and it was so different to anything I'd ever seen before.
Was it a double take moment? Or was it...
Yeah, actually, I think I had to look twice. But no, it was quite amazing. Yeah, I don't know. You hope when you do your research, you hope you have the right hypothesis. You know, you really want to make a difference. But when you actually see it come to fruition, that moment is the best. You know, it takes away all the challenges that you may have had, whether you're writing grants or you've had your paper rejected, or, you know, your funding didn't get up. When you get that moment when, you know, you could make a difference to someone's lives because of scientific discovery, it's worth it.
Still that ending on a high.
Mm. Oh,
Was it-
Can I just pop a question in, Stuart?
Sorry?
Can I just pop a question in?
Okay, well, Senator Palmer, what's the question? But just
Just
feed it
looking
through me.
What frustrates you the most of the current research technologies?
frustrates me the most. Is it do you mean the funding?
Stu.
Yeah, I will look at him.
Yeah, the whole. Just, I mean, what do you find
In
most
general.
frustrating about conducting your work?
Yeah. Hey? The people that walk in the corridor when we're trying to film the thing, the people that walk past the corridor, and yeah. I mean, okay. You know, one of the frustrating things at the moment is we've got some brilliant minds doing research, but we just don't have enough money to fund everyone. And so there's some fantastic research that's just not being funded. So, you know, best case scenario would be to increase the funding in Australia by tenfold, you know, so we could really support these fantastic advances. And Australia does excellent research. You know, the impact of our research is huge. So I think if we could, yeah, I don't, could that end there? The other thing is, I guess, we've got all this massive equipment. We're interrogating all of these. I'm just saying this. I know this is not big, but we really need the money to support the personnel and the bioinformatics and the, you know, multidisciplinary people to come in at the end and actually be able to analyse all this data we're getting. What does it mean? Like, what can we do with it? So we're almost generating all this data, but we need to know what are we going to do with it. So for me, that is a little bit frustrating because cancer is so heterogeneous, which means it's different in every person and it's even different within each person, whether depending on whether it's growing. There's a lot of information that you get from an individual's cancers. And what you need to use that information for is very challenging. Which bit of that information is important in targeting for a therapy? which cancer cell will actually resist the therapies that that patient is currently receiving and needs to be targeted individually. So it's a really tricky situation, actually gathering all this information, but knowing how to use the information in the best way.
More information than analysis.
Yeah, I mean, it's becoming like that. We're doing a lot of information gathering, and heterogeneity, is huge. So I guess by gathering more and more information, we're trying to find some similarities that we could target.
When we first did the proposal, Ben and I thought it was a bit like Enigma in World War II and building Colossus.
Yeah.
To crack the Enigma code with the Germans, that was uncrackable. It sounds like you're getting the information, but you're Turing as well.
Yeah.
You're Alan Turing trying to figure out.
I think there's two types of information. There's, the code, the genetic code, for example, but then there's this plasticity, what we call plasticity, how the cancer cells change, constantly changing, and the ability to actually interrogate that. How are they changing? What are they interacting with to allow that change to occur? How do we block those interactions? That's so fascinating, but it's also where we need to go, I think, for targeting metastatic disease.
Be a moving target.
Yeah. I mean, if you can, for example, bone metastasis is the most common site in, solid cancers. So if we could target the very earliest steps of bone spread, could we then block the ability of cancers to go to the lung and the liver and other places where, patients can decline rapidly when they get those metastases? What we want to do is target those steps that are early, that could then lead to a cure. And that would be ideal. Yeah.
Beautifully said. I don't have any more questions unless there's something you'd like to ask or something you'd like to say.
I didn't talk about the ADHD, but I don't think you'd probably use it anyway, would you?
The what?
The ADHD, I don't think you'd use it. It's a bit,
Your ADHD.
yeah.
We'll just put a super up there.
Large red letters.
Noticed?
BSc,
Yeah.
PhD, ADHD.
Put it as when everyone else has, OA,
FRC.
I'll have
If you're
ADHD.
military, because when you get into it's ADHD and bar.
I just hope I haven't said me too much because it's my team that's been important. You don't think I've done that?
I think we would have pulled you up.
Sounded all right? I think I just wanted to get the bone thing across. We got that across. We got multidisciplinary across. We got everyone's cancer's different.
Why? Like if you're going to metastasize, bone sounds like a really hard place to end up, whereas soft tissue nearby would be a much shorter journey.
Well, the lymph node is an easier journey, but
Yeah.
that's in the lymphatic system.
Yeah.
Sorry. So, a common first site of metastasis or spread is the lymph nodes. Then that gives you some idea about the characteristics of the cancer and potentially the risk that it may come back in distant sites. But it's really the spread to distant tissues such as the bone that becomes the deadly sites. And the function of different cells in that process and the biology behind it is really important, because even though we are getting better at diagnosing earlier, at treating patients earlier and eradicating metastases if we're very early, some patients will still develop spread to distant tissues, and we absolutely need to be able to target those to prevent all deaths from cancer. So that is a critical area that I'm very interested in. I don't think I answered your question. I just went off on another tangent.
It was a good tangent.
This is more like lymph makes sense because it's kind of a highway system.
Yeah.
So you're in there. Is it? Do you ever think cancer can think? Like,
Can what?
Like, it sounds like it's conscious. I mean, obviously, is the way cancer behaves. It just seems to.
It's not that it's thinking, it's being selected for. So, for example, bone is the most common site of spread and harboring these dormant cells, because what cancer cells do, okay, so It's actually very interesting. So bone is one of the most common sites of cancer cell spread or dissemination because these cells can actually sit in areas that are called immune privileged. So in the bone, there are little stem cell niches, they're called, that are immune protected. So a cell can sit there and not get targeted by the immune system. The cancer cells use that. They go and sit there as one of their mechanisms of how they survive in that environment. Also, the cancer cells then utilize other cells in that tissue that become really important for their growth, and that's the bone cells themselves. You know, by degrading the bone, they release growth factors that allow them to grow bigger. By making more bone growth, they actually also change the environment to support them. So, you know, some of these sites that the cancers go to is because they're being selected for in that environment because the other normal cells are helping them. Yeah.
So it sounds a bit like if the cancer cells are seeds blowing in the wind
They're like seeds.
around the body,
Yeah.
a lot of them are just, they're not going to thrive where they land. Whereas those ones have landed in exactly the right spot to actually
Yeah.
do well. So it's like fertile soil. They're not
Exactly what
initially
it's called,
true.
seed and soil hypothesis. Have you heard of that? Have you heard of the seed and soil hypothesis? It's funny because they do talk about the seed and soil. Yeah. Yeah.
So it's from dandelions to sycamores.
Yeah. Yep. So cancers can be like seeds, and there is a seed and soil hypothesis. Why do some cancer cells go to certain sites, like the bone? And it's thought that they find a fertile soil. So a little bit of it is, where does a cancer cell sit? Where does it arrive? What is around that environment to support its growth? And so, you know, certain environments will be able to contribute to that growth more than others. So a little bit of is I guess, luck for the cancer cell, where it's sitting. Is it sitting in a site where it will be immune protected? Can it actually recruit other cells to come in? So what is the nature of that cancer cell? Can it secrete other proteins and say, come along and help me? And that is what actually occurs in metastatic tissues. And so the interaction between a cancer cell and other cells in that environment is critical in whether it will become a large cancer that is problematic.
And it depends on how it's broadcast, that seed and soil. I mean,
Yeah.
Which method is used to be broadcast across the body, or now it's housed.
Yeah.
Even. Good luck with that.
No, metastasis is a really difficult area. It's very difficult to study because actually making preclinical models to mimic that really interesting yet very complex stages of metastasis is quite difficult. But it's really important to study, and so that's why we're developing new models at the moment, so we can say, okay, would what we think work in a preclinical model actually work in a patient? And that difference is very important. We don't want to go towards a clinical trial until we have this peace of mind that what we've found in the laboratory could actually change the state of a cancer that we get directly from a patient. Yeah. Was that ringing for long? I didn't notice.
No,
No, so, right,
That's hyper-focusing.
so I got in under the line, didn't we?
We did.
I'm happy.
That's a great way of describing
The seed and soil. It's funny that you said seed because that's what the whole hypothesis is, seed and soil. It's Paget's hypothesis.
See what a dandelion, blowing a dandelion,
Just
a lot of seeds.
cut.
Think about what I'm going to say now.
Speaking. Professor Belinda Parker.
Okay, I'm just going to think. You know,
There's no rush.
I just have like a message for cancer patients. We are getting so much better at treating and personalising therapy that you're in good hands. You know, a lot of patients that I know, they spend their life stressed about what type of cancer do I have? Will the cancer come back? And I'm hoping from the research and from the trials that they can get to a place where they're not as stressed. We can tell you what type of cancer you have. You know that you're getting the best treatment you can, and to take away some of that, you know, uncertainty for these patients. Did that sound shit?
I should pull at the end, Mrs. Derek.
I don't want you guys to look at those bits. Make sure you get rid of them because I do pull terrible faces. Because I find myself so cringy.
We all
Yeah.
find ourselves cringy. I mean, especially in
Self-cringing.
Oh, did you? Well, you don't. Anyway, if it looks cringy, you don't have to.
I know all sorts of people who've got cancer and they say that's it, I'm going to die. It's like, well, are you? Do you know? And they say, well, I don't know.
I actually have. I actually say to my friend who's my age that has, actually she's a couple of years younger than me, that has breast cancer that was quite aggressive when it was diagnosed. I say, she always goes, I think I might have bone. I go, you may as well have bone metastasis because you're living your life like you've got it anyway. Like and she actually, I wouldn't say that to someone I didn't know, but she said that actually made a difference for her because she was living it like a death sentence. And so, what's the point in waiting for it? Obviously, I gave her the stats and that she had a really good response to her therapy too. But
And does thinking make it so?
yeah, I mean, it's easy to say. I think, everyone would be similar, right? It's easy to say to someone else when you don't have it yourself. So, yeah, but anyway. But that's what happens. It's like on their mind constantly, is when they get aches and pains.
Which is debilitating in itself.
It is, and it's not talked about as much. It's just like, give them these therapies, and
One thing we talked about doing is a book called, I think someone's already done this book called Fuck Cancer.
yeah,
It would just
heard
be
about it.
getting some really cool designs on the thought that kept people going. And a mate of mine had esophageal cancer.
Oh,
which
yeah.
is generally
Yeah.
pretty bad.
Terrible.
But he got a